Good morning and good afternoon, ladies and gentlemen, and welcome to our symposium, which is about hypoxic pulmonary hypertension, a possible complication of COVID-19 pneumonia. As an introduction, pulmonary hypertension is associated with increased mortality and morbidity in the world. Pulmonary hypertension may occur by different reasons, and one of them may happen in COPD patients. There is a general agreement that chronic hypoxia plays a central role in generating pulmonary hypertension in COPD patients. However, neither the underlying mechanisms nor 100% efficient treatment are available for COPD patients with pulmonary hypertension so far. On the other hand, more than 242 million of COVID-19 cases have been reported with more than 4.9 million deaths around the world. Despite many progresses in the treatment of COVID patients, still there is no exact therapeutic approach, in particular in severe cases of the disease. What is clear is that alveolar and arterial hypoxia is the important complications of the disease. As we know, alveolar hypoxia leads to a physiological phenomenon of hypoxic pulmonary vasoconstriction. However, the increase of pulmonary vascular resistance may have a negative effect on right ventricular function by increasing the afterload. Besides, the question remains to be answered whether or not the prevalence of pulmonary hypertension increases in the patients with severe COVID-19. In this symposium, we have invited specialists in clinical and basic sciences to discuss about these issues. First of all, I cordially appreciate all invited speakers for their time and sharing their experiences with us in the program. I also thank all participants for taking part in the program and their time. Now it's my pleasure to introduce Professor Masumpour, his fellowship in pulmonology and the head of the Department of Lung in Faghi Hospital at Shiraz University of Medical Sciences. He is also advisor of the Research Chancellor of Shiraz University of Medical Sciences. His lecture will be presented in Persian. You can start, please. جناب آقای دکتر معصومپور، لطفاً سخنرانی خودتون رو بفرمایید.
ممنون خانم دکتر. به نام خدا با سلام خدمت اساتید و همکاران گرامی و تشکر از اینکه به من این فرصت داده شد. من انشالله یه پرزنتیشنی در مورد منیفستیشن های کلینیکال کووید خواهم داشت، حالا با یه تمرکز بیشتر روی بحث پولموناری که اسلایدها رو با اجازه هیئت رئیسه محترم شروع میکنم. انتهای سال 2019 بود، همونجور که همه دیگه میدونن و چندین بار تکرار شده که دسته‌هایی از تظاهرات تنفسی بیماران بروز کرد که نهایتاً منجر به پیدا کردن یک کرونا ویروس جدید شد که توی ووهان چین بود و خیلی سریع کل دنیا درگیر شد. الانی که داریم با هم دیگه صحبت می‌کنیم، همونجوری که خانم دکتر فرمودن، آمار و ارقام رو تعداد 242 میلیون نفر در کل دنیا درگیر هستند و نزدیک به 5 میلیون فوت متاسفانه از این بیماری داشتیم و این نگرانی و دغدغه وجود داره. ما همیشه توی این فصلی که الان هستیم، پاییز و زمستان، افراد معمولاً بین 2 تا 3 بار یه سرماخوردگی‌هایی به هم می‌زدن و خب با یه استراحت خیلی کوتاه مدت یا حتی بدون استراحت هم کاندیشن در حقیقت استیبل میشد و مشکلی وجود نداشت. ولی الان هر کسی که 1-2 تا سرفه بکنه و یه عطسه بکنه، همه با یه نگاه خیلی شک آلودی بهش نگاه میکنن و نگران هستن که آیا ایشون کووید رو دارد یا نه. پس یه ظن بالینی خیلی مهم نیاز هست که داشته باشیم با توجه به بحث پاندمی. الان هر بیماری که میاد با علامت‌های کورایزا، تب، بدن درد، تقریباً almost always ما تشخیص اولیه‌مون برای این فرد کووید مطرح خواهد شد و تمام کسایی که مشکوک به کووید هستیم برای اینکه بتونیم کنترل بکنیم این پاندمی و اپیدمی رو قاعدتاً باید یه تست آزمایشگاهی انجام بدیم که چویس برامون انجام PCR هست. ولی یه نکته وجود داره، ما معمولا توی کلینیک وقتی که یک نفر از یه خانواده تستش مثبت میشه و بقیه علامت دار هستند، ما حتی اگر تست اولازم منفی بشه از این فردم چکش بکنیم، عادتا میذاریم به حساب در حقیقت فالس نگاتیو و اون پرکاشنای مربوط به قرنطینه و رعایت و اینا رو حتما توصیه میکنیم و موارد دیگه ای که حالا در ادامه بحث در موردش صحبت خواهیم کرد. یه نکته که کارو سخت میکنه بحث ایسیمپتوماتیک بودن بعضی از افرادی که این ویروس رو دارن حمل میکنن ولی علامتی ندارن. ریپورت هایی که داریم عددا مختلفه، تا 40% گفتن که امکان داره که موقعی که این ویروس به سیستم بدن وارد میشه علامت خاصی ایجاد نکنه، یه علامت انقدر کم باشه که فرد متوجهش نشه. از این نظر برام مهمه چون این فرد میتونه چندین نفر دیگر رو درگیر بکنه. و نکته جالب اینه که وقتی که این افراد حتی ایسیمپتوماتیک هستن، دیدن افرادی که توی تست در آوردن که اینا تستشون مثبت هست، بررسی کردن دیدن تا 50% موارد اینا امکان داره درجاتی از درگیری توی ریه به صورت گرام گلاس رو داشته باشن. با این توضیح که الزاماً این حد کم از درگیری شاید تغییر توی سچوریشن اکسیژن ایجاد نکنه، شاید سرفه ایجاد نکنه و علامتی هم نداشته باشه. از این نظر کار یه مقداری سخت میشه. حالا تو قسمت ایمیجینگ من یه مقداری روی این نکته بیشتر فوکوس میکنم و توضیح خواهم داد. و نهایتاً این افراد ایسیمپتوماتیک یا افرادی که بگیم حالا در حقیقت ویروس وارد بدنشون شده، یه درصدشون نهایتاً این حالت هست که منجر به علامت میشه. مدین تایمی که هست یه چیزی هولوش 3 روز 4 روز 5 روز هست که اکثر مریضا ما موقعی که یه فرد میاد دغدغه داره میگه من توی جلسه‌ای بودم، توی یه جایی بودم که یک نفر تستش مثبت شده، ما میدونیم که تا حدود 14 روز این احتمال وجود داره که از نظر کلینیکال خودشو پرزنت بکنه و در مواردی شاید حتی طولانی‌تر، ولی عمدتاً توی 3 4 5 روز اول اگر بخواد بروز پیدا بکنه بروز پیدا میکنه. و خوشبختانه درصد زیادی از اینا اگر هم سیمپتوماتیک بشن فرم مایلد خواهد بود که خیلی جای نگرانی نداره، حتی بدون هیچ مداخله مدیکالی هم اینا وضعیتشون کنترل خواهد شد و هیچ مشکلی هم تهدیدشون نمی‌کنه. نکته مهم سن فرد و وجود کوموربید دیزیزها هست که بیشتر در موردش صحبت می‌کنیم که یه مقداری تمرکزمون رو مشخص بکنیم. حدود نزدیک به 80% مریضامون فرم مایلد خواهند داشت که یا نمونیا ندارند یا بسیار بسیار خفیف هست که برامون از نظر کلینیکال شاید سیگنیفیکانس نداشته باشه. اهمیت این افراد فالو کردنشون هست که قاعدتاً این افراد باید دنبال بشن، روزانه از نظر تب و میزان اشباع اکسیژن فالو بشن، از نظر تنگی نفس و سیمپتوما که آیا به سمت بدتر شدن پیش میرود یا نه. فرمایی که خیلی ما نگران میکنه ما رو فرمای شدید هست که موقعی که درگیری ریه ها بیشتر از 50% باشه یا همراه با هایپوکسیا و دیسپنیا باشه که خب اینا معمولاً یه درصد هولوش 10 تا 15% رو شامل میشن و اونایی که کریتیکال میشه حدود 5% هستن، یعنی اونایی که نهایتاً کارشون به ICU و مکانیکال ونتیلیتور میکشه حدود 5% میشه. اوورال مورتالیتی هم یه چیزی هولوش 2.3% هست که از نظر حالا یه بیماری شاید عدد زیادی نباشه ولی با توجه به اپیدمی و اندمی و این وسعت درگیری عدد و رقم بسیار بزرگ هست و حتی یک مرگ هم فاجعه‌ای توی یک خانواده ایجاد میکنه که واقعا مدیریت کردنش بسیار سخت است. اکثرا افراد الان تو سنای میدل ایج و جوان هستن. حالا تو پیکای اول دوم ما مورتالیتیمون عمدتاً توی افراد با سنای بالاتر بود ولی الان توی پیکای اخیر متاسفانه سن مورتالیتی هم یه مقداری پایین‌تر اومده که بسیار حائز اهمیته. اینجا اون فیتالیتی ریتی که داره بر اساس ایج رو مرتبش کرده و همونجور که می‌بینیم از سن تا حدود سن 10 سال حدود 2000% ما مرگ داریم تا میاد بالای 90 سال که 25% به بالا میتونه مورتالیتی داشته باشه که خب این اعداد و ارقام توتالی هست و مربوط به جامعه خودمون نیست. از نظر عدد و ارقام توی جوامع مختلف موقعی که هرمای سنی رو میبینیم شاید بشه با تصحیح کردن اون هرم سنی عدد و رقم رو به دست بیاریم که یه تحلیل جالبی یکی از همکاران اپیدمیولوژیست داشت در مورد مرگ توی بعضی از کشورها و مقایسهش با مثلا ایران که نهایتاً یکی از آیتمایی که توضیح داده بود گفته بود که چون هرم جمعیتی ها متفاوت هست بعضی وقتا توی یه جمعیت افراد مسن بیشتر یا افراد میدل ایج بیشتر، خب این میتونه تاثیرگذار باشه توی خروجی که نهایتاً منجر به اون مورتالیتی که توی آمارا داریم بشه. و یه بحثی هم بود همیشه، الان دوباره فصل پاییز هست و بحث آنفولانزا، همیشه میمدن مقایسه میکردن، یکی دو سال پیش حتی میگفتن خب نه این کرونا ویروس هم شبیه آنفولانزا هست، خیلی مهم نیست، خیلی مسئله نداره، ولی نهایتاً دیده شد که کووید یه چیز هولوش 5 برابر مورتالیتیش موقعی که کار به هاسپیتالیزیشن میکشه از آنفولانزا بیشتره. یعنی یه چیزی عدد و رقم خیلی زیاد تفاوت میکنه و این حالت توی کشورهایی که از نظر ریسورس و از نظر منابع و از نظر بیمارستان و استاف دچار مشکل هستن به مراتب نمودش خیلی وحشتناکتر میشه و اوضاع خیلی خرابتر میشه، طوری که مثلا توی گزارش‌ها هست که مریضایی که این هاسپیتال بودن مورتالیتی تا 30 روزشون تا 48%، اونا که کار به آی سی یو ادمیت کشیده یعنی نصف مریضا عملاً اکسپایر میشدن و این خیلی عدد و رقم بسیار بالایی هست که برامون اهمیت قضیه رو میرسونه. مورتالیتی اسوسییشن دیگه هم با کوموربید دیزیز داره که در مورد کوموربیدام حالا صحبت میکنیم. سن رو که من عرض کردم چند تا اسلاید قبل‌تر، کوموربیدایی که برای اون مهم هست کاردیوواسکولار دیزیزا انواع و اقسام، بحث هایپرتنشن هم شاید بشه تو همینجا جا داد، دیابت، بیمارانی که سی او پی دی و آندرلاینگ دیگه ریه دارن، دیستراکتیو لانگ دیزیزایی که وجود داره، کنسر به خصوص هماتولوژیک مالگنانسیا، لانگ کنسر و اونایی که متاستاتیک هستن، بیمارانی که سی کی دی هستن، کرونیک کیدنی دیزیز دارن و اونایی که ارگان ترانسپلانت شدن، بیمارانی که اوبس هستن و اسموکینگ. در مورد اسموکینگ یه کانتروورسی هم حالا یه بحثی روش هست که میگن بعضی وقتا اون در حقیقت افراد اسموکر کارشون به هاسپیتالیزیشن کمتر میکشه، شاید بیماری بگیرن ولی هاسپیتالایز کمتر میشن. یه دو تا ریویو در حقیقت سیستماتیک ریویو هم روی این قضیه هست، ولی خب این بحثش بازه هنوز. الان اگه از کسی سوال بکنیم که اسموکینگ آیا به عنوان ریسک فاکتور سیویر ایلنس هست یا نه، باید بگیم بله، به خاطری که هنوز اون نهایی نشده و اوبزیتی هم که واقعا وجود داره و نشون داده شده که فرد میتونه دچار مشکل بشه موقعی که اوبیس باشه. فاکتورهای دیگه سوشیو اکونومیک بک‌گراندها هست، به خصوص تو جوامعی که حالا نژادهای مختلف هستن. ریپورت‌هایی که از آمریکا هست، نژاد بلک و هیسپانیک با درگیری بیشتر و مورتالیتی بیشتر هستن. شاید کوالیتی کیری که بهشون داده میشه متفاوت باشه و یا فاکتورهای ژنتیک بتونه دخیل باشه. بحث جنسیت که توی آقایون به نظر میاد اوورال کریتیکال کیس ها و دس بیشتر باشه به نسبت خانم ها. و بحث دیگه بحث خود فاکتورهای مرتبط به خود ویروس هست، اگرچه رابطه 1:1 و 100% خطی نیست، ولی هر چی که لود وایرال آر ان ای بیشتر باشه به نظر میاد که سیویریتی دیزیز بیشتر باشه و دیدن بیمارانی که تونستن وایرال آر ان ای از خونشون جدا بکنن اینا نهایتاً اسوسییشن با ارگان دمیج ها بیشتر بوده. یعنی اگه کسی تست در حقیقت آر ان ای ویروس توی خونش مثبت بوده، درگیری ریه، قلب و کلیه توش بیشتر بوده. از نظر ژنتیک هم کارهای متعددی انجام شده. یه بحث گروه خونی هست که حالا یه مطالعه بود که من دیدم که نوشته بود مثلا گروه خونی تایپ او یه مقداری خطر درگیری و خطر سیویریتی توش کمتره، در مقابل گروه خونی A بیشتره. البته هیچ کدوم از اینا به صورت دیفینیت و 100% شاید واقعا نشه چون کیسای متفاوتی رو ما می‌بینیم. این یه تخمین کلی هست از بابت این قضیه. ما موقعی که توی کلینیک هستیم اولین کاری که می‌کنیم از مریضمون تاریخچه خواهیم گرفت و معاینه‌ای از بابت صداهای ریه خواهیم داشت. تمرکزمون روی میزان اشباع اکسیژن خواهد بود و تب بیمار که به صورت کلی این کار انجام خواهد شد. معمولاً همونجور که عرض کردم در عرض 2 هفته بعد از اکسپوژر اگر فرد بخواد علامتی بروز بده، قاعدتاً بروز میده که عمدتاً هم توی 4-5 روز اول خواهد بود، همونجوری که صحبت شد. معمولاً علامت به صورت ضعف و بیحالی، سرفه و سردرد، اینا 2-3 تا علامت بسیار شایع هست. حالا درصداشو توی اسلاید بعد می‌بینیم. علامت‌های گوارشی به صورت حالت‌های درد شکم و حالت‌های اسهال، اینا میتونه وجود داشته باشه و تغییری که توی حس چشایی و بویایی هست. اینا هیچ کدومش به صورت 100% نیست، چون بعضی از مریضا مثلا کلینیک میاد علامت‌ها همه تیپیکه، میگه خب آقای دکتر چون من بویایی من هنوز دارم پس احتمالاً کرونا نیست. یعنی بعضی از استنتاجایی که افراد از اون برای قضیه میکنن جالبه و با همین استدلال میگه من مشکلی ندارم، سر کارش میره، تو محل رفت و آمد میکنه و متاسفانه باعث درگیر کردن افراد دیگه میشه. اما نگران کننده ترین پرزنتیشن بحث ذات‌الریه یا همون نمونیایی هست که میتونه فرد دچارش بشه. تقریبا اگر نگیم 100%، تقریبا نزدیک به 100% کسایی که نهایتاً ریه درگیر میشه و درگیری هم وسعت پیدا میکنه کسایی هستن که شروع بیماریشون یه شروع طوفانی هست، یعنی با سرفه و با تب و لرز و خیلی حالتهای خیلی اکیوتی هست. اینا خیلی بیشتر نمود داره تا اینکه اگه گراجوال باشه عمدتاً در همون حد اگرم درگیر میکنه خیلی دیگه پروگرسی نداره. حالا تو قسمت لب دیتا یه مقداری من راجع به فاکتورایی که شاید بتونه کمک بهمون بکنه برای دسته‌بندی بیماران من یه نکته کوچولویی هست خدمتتون عرض میکنم. این اسلاید پرزنتیشن ها رو میگه تا 50% نصفشون میتونن با سرفه بیان، تب بالای 38 توی 43%، ملیز، هدیک، ترتیبا رو دیگه میبینیم و نهایتاً که ما یه چیز اولوش 10% افراد شاید حس بویایی چشاییشون دچار مشکل بشه یا علامت‌های راینوره داشته باشن یا ابدومینال پین داشته باشن. پس خیلی روی اینا هیچ کدومش به صورت تکی حساب نمی‌کنیم و کل پکیج رو به صورت کامل در نظر می‌گیریم و برای مریضمون اون ظن بالینی رو مطرح می‌کنیم. آزمایش‌هایی که توی پاراکلینیک به ما کمک می‌کنه لمفوپنی، افت تعداد لمفوسیت‌ها هست، در مواردی افت پلاکت هست، افزایش آنزیم‌های کبدی، افزایش LDH، فاکتورهای التهابی مثل سی ریاکتیو پروتئین، فریتین، سایتوکاین‌های التهابی مثل اینترلوکین 6، TNF آلفا، افزایش دی دایمر، افزایش پرلانگ شدن PT و در مواردی هم که حالا درگیری‌هایی داشته باشیم از نظر سیستم کاردیوواسکولار، تروپونین میتونه رایز پیدا بکنه یا CPK مریضمون. دیده شده اگر یه مریضی پرزنتیشن اولش با تب شدید نباشه، علامتاش هم خفیف باشه، فاکتورهای التهابی من جمله CRP یا ESR حالا اگر که چک بکنیم بالا نباشه، کورس نسبتاً اسموتی رو طی بکنه و خیلی فراز و نشیب نداشته باشه. حالا بیشتر راجع بهش صحبت می‌کنیم. ایمیجینگی که ما ازش کمک می‌گیریم یه واقعیتی که الان واقعیت تلخی که توی جامعه ما هست اینه که الان هر کسی که اولین شک و ظن بالینی رو در مورد کووید براش مطرح میکنن سریع 1-2 تا CT اسکنر شده تا بخواد برسونه خودشو به یه پزشکی که حالا بخواد تصمیمی براش بگیره. اکثر مریضا سراسیمه توی کلینیک‌های رادیولوژی هستن و دارن این اتفاق میفته که نگرانی ما برای چند ماه و چند سال آینده هست. بحث اشعه‌ای که این بیماران دارن میگیرن مخصوصا توی خانم‌های جوان خیلی نگران کننده خواهد بود. اگر که ما مریضمون شدید نباشه، سچوریشن خوب باشه، معاینه خوب باشه، فاکتور التهابی بالا نباشه، حتی اگر هیچ ایمیجینگ هم انجام ندیم مشکلی اتفاق نخواهد افتاد. ولی اون دغدغه‌ای که بیماران دارن معمولاً منجر به این میشه که پزشک اول مقاومت بکنه، پزشک دوم، پزشک سوم میگه آقا برو یه CT اسکن انجام بده بیا، و همونجوری هم که خدمتتون توی اسلاید دوم سوم گفتم و نیمی از افراد اسیمپتوماتیک هم بالاخره درجاتی از درگیری رو دارن. بعد طرف همین CT رو برمی‌داره میاره توی کلینیک یا توی مطب میگه تو نگفتم برام یه CT اسکن بنویسین، الان بیاد دیگه من درگیر شده، حالا باید چیکار بکنم؟ در صورتی که همه درگیری‌ها به سمت بدتر شدن پیش نمیره. حالا اگه قرار شد یافته داشته باشه چیست؟ عمدتاً به صورت گراند گلاس اوپاسیتی یا کانسالیدیشن هایی است که معمولاً دو طرفه هست، محیط پریفری بیشتر درگیر میشه و قسمت‌های تحتانی ریه بیشتر از قسمت‌های ایپکس درگیر میشه. زمان درگیری، پیک درگیریمون عمدتاً روزهای 10 تا 12 هست، یعنی میشه انتهای هفته دوم شروع علامت‌ها اگر مریضی بخواد مشکل به هم بزنه. درصد بسیار کمی از اینا امکان داره اسپانتانیوس نوموتراکس به هم بزنن که توی ریپورتی که بوده 56%. عددش بسیار کمه، حالا به خاطر کوموربید دیزیزا هم شاید باشه، افتراقش یه مقدار کار سختی هست، ولی بالاخره این حالت میتونه رخ بده. یافته‌هایی که تو سیتی هست در 83% موارد که فرد یافته داره میتونه گراند گلاس اوپاسیتی باشه، به صورت میکس با کانسالیدیشن توی نزدیک به 60% موارد، تیکنینگ پلورا توی 52%، ولی پلورال افیوژن یعنی تجمع مایع توی فضای پلور چیز خیلی شایعی با کووید نیست، مگر که کامپلیکیت شده باشه. خود ضخیم شدن اینترلوبولار سپتاها هست که تو نزدیک به 50% موارد، و ایربرونکوگرام هم که میتونه توی نزدیک به 45-6% موارد بروز پیدا بکنه. حالا اگه ما نخوایم از اشعه به مریضمون بدیم، میتونیم از چه مدالیتی دیگه کمک بگیریم؟ اکسپریانس باش زیاد نیست، ولی سونوگرافی ریه ها هم جایگاه خاص خودش داره که توی یه سیستماتیک ریویو نشون داده که یه حساسیت حدود 86% و ویژگی 55% داره. ویژگی این مقداری پایین هست، ولی حساسیت قابل قبول هست در مواردی که بخوایم فالو بکنیم با یه فرد رادیولوژیست که اکسپریانس خوبی داشته باشه، شاید بتونه یه مدالیتی بینابین باشه از هیچی انجام ندادن تا رفتن سراغ سی تی اسکن. حالا چیزایی که تا اینجا بالاخره فرد با ویروس درگیر شد و یه سری مسائل مشکلاتی داشت، حالا اگر خفیف بود که عمدتاً 80-81% موارد خفیف هم هست، مایلد هست، هیچ اتفاقی نمیفته. ما نگرانیمون برای اون 20% باقی مانده است که چه چیزایی تهدیدشون میکنه. این کامپلیکیشن هایی که توی 2 تا اسلاید آخر با هم دیگه مرورش میکنیم. بحث نارسایی تنفسی که برای این افراد اتفاق خواهد افتاد که همون بحث ARDSی که میتونه ایجاد بکنه برای فرد، و نهایتاً هایپوکسیایی که هست که حالا فوکوس و تمرکز عمده این لکچرها و سری لکچرها رو همین داستان خواهد بود. و دیده شده که نزدیک به 12 تا 24% افرادی که نهایتاً هاسپیتالایز میشن به خاطر این داستان و هایپوکسیک هستن، کارشون نهایتاً به مکانیکال ونتیلیتور کشیده میشه. آماری که ما تو آی سی های خودمون داریم، حالا از دوستانی که در حقیقت کار ویزیت این مریضا رو انجام میدن، ما اول فکر میکردیم دوستانی که شیراز ویزیت میکنیم گفتیم شاید مورتالیتی ما واقعا عدد و رقم بالایی باشه. اون کسی که کارش به آی سی یو و مکانیکال ونتیلیتور میکشه تا 70-80% موارد متاسفانه منجر به فوت بیمار میشه. آمار خیلی بالاست، ولی تمام کیرایی که اول ما مقایسه‌ای که کردیم گفتیم شاید حالا استاندارد کیرا متفاوت باشه فلان باشه، تقریبا تمام کارهایی که انجام میشد دیدیم نه، با اجاست کردن همه اینا باز مورتالیتی عددش بسیار بالاست. در کنارش کامپلیکیشن های کاردیاک و کاردیوواسکولار هست که به خاطر وجود اون هایپرکوابیلیتی استیتی که توی ستاپ خود کووید اتفاق میفته که توی اسلاید بعدم داریم، میتونه عوارض و کامپلیکیشن های مختلف و متعددی ایجاد بکنه و بعضی وقتا مایوزایتیسی که توی قلب ایجاد میکنه خودش اریتمیا و نهایتاً منجر به نارسایی قلبی و یا شاک استیت میتونه بشه برای بیمار. اون ترومبو امبولیک فنومنا که هم قسمت ونوس و هم قسمت آرتریال که تو قسمت ونوسش ما عمدتاً حالا بیشتر میبینیم، آرتریال هم هست مثل به صورت اکیوت استروک و ایسکمیا بروز میکنه، ولی اون مشکل پی تی ای یا آمبولی ریویش هست که باز خودش میتونه پولمونری هایپرتنشن رو در حقیقت ایجاد بکنه و فشار رو اونجا بالا ببره. کامپلیکیشن های نورولوژی که به صورت انسفالوپاتی و استروک که حالا میتونه رو زمینه خود اون ایونت قبلی که توضیح دادم یا به خاطر خود التهابی که توی بافت مغز ایجاد میشه میتونه ایجاد مشکل بکنه. موومنت دیزوردرها، اتاکسیا و سیژر که نادرتر میتونه دیده بشه. کامپلیکیشن های اینفلاماتوری که به صورت افزایش سایتوکاین ها و افزایش مارکرهای التهابی هست که روی همین زمینه مطرح شد بحث داروی توسلیزوماب که میتونه کنترل بکنه در حقیقت اینترلوکین 6 و در حقیقت رسپتور بلاکر اون باشه. عفونت‌های ثانویه تو بحث کووید اونقدری که ما در موردش در حقیقت نگران هستیم و آنتی بیوتیک میدیم و آنتی فانگال میدیم شاید اینقدر زیاد نباشه. توی یه رپورتی که از چین بود حدود 8% و رپورتی که اروپایی‌ها کار کرده بودن حدود 4 تا 5% سکندری اینفکشن رو مطرح کرده بودن و میشه با ادجاست کردن داروها و تعدیل مصرف کورتون و داروهای دیگه شاید درصد زیادی از این کامپلیکیشن های ثانویه و سکندری اینفکشن ها رو کنترل بکنیم. در مورد ریکاوری و اون عوارض طولانی مدتی که بیماران دارن، خوشبختانه حالا توی 1.5-2 سال گذشته که درگیر بحث کووید بودیم، اکثر مریضا که حالا با کپسول اکسیژن از بیمارستان ترخیص میشن در عرض 1 تا 2 ماه معمولا اینا میتونن ایندیپندنت از اکسیژن و یعنی نیزال دیگه استفاده نکنن و کاندیشن کم کم برگرده، هرچند که ویکنس اینا تو 4 تا 6 ماه امکان داره ادامه داشته باشه و اون فرد قبلی خیلی سریع نشه، ولی دیگه حداقل دیپندنت نیست، کار شخصیش رو میتونه انجام بده و این خودش یه قسمت خوب داستان هست، به جز مواردی که نهایتاً منجر به لانگ فایبروسیس میشه و کامپلیکیت میکنه که درصدش بسیار بسیار کم هست. بیمارانی که فرمای خفیف تا متوسط رو دارن در عرض 2 هفته ما اون بحث قرنطینه رو میتونیم طی بکنیم و کاندیشن مریضمون خوب باشه. اونایی که شدیدتر باشه یه مقداری تایم قرنطینه رو طولانی‌تر میکنیم، هولوش مثلا تا نزدیک به 1.5 ماه و عمدتاً هم همونجور که عرض کردم معمولاً توی یه روند 2 تا 3 ماه دیگه اینا اکثراً برمیگردن. یه سری عوارضی امکان داره بمونه مثل حالتهای ضعف و بیحالی، دیسپنیا، چست پین، سرفه‌های زد و گذری، اینا خیلی نگران کننده نیست و خیلی اوضاع رو از کنترل خارج نمی‌کنه. من سعی کردم خانم دکتر خیلی سریع مطلب رو جمعش بکنم، حالا اگر کامنتی دوستان دارن یا سوالی هست من خدمت همکاران و اساتید هستم.
خیلی ممنون آقای دکتر به خاطر پرزنتیشن عالی شما.
سلامت باشید.
اگر شرکت کنندگان محترم سوال دارن میتونن با چت یا میتونن درخواست بکنن که میکروفونشون رو آن بکنید و سوال خودشون رو مطرح بکنن. ظاهرا سوالی مطرح نشده.
آقای دکتر نجف پور کامنتی دارین آقای دکتر؟
عرض سلام دارم خدمت شما آقای دکتر. سوال من اینه که ما خیلی شنیده بودیم که وقتی که آلوالای ریه حالا از مشکل پیدا میکنه از بین میرن یا دیوارهای بینشون چی میشه از بین میره، اینا بازگشت ناپذیرن، یعنی ظاهرا سلولاشون طوری هستن که این مسئله که در کووید اتفاق میفته به این کامپلیکیشن ها تا چه حد اینجوری هست و این که فرد بعد اون که بیماری چی میشه بهبود پیدا میکنه در ظرف حالا 2 ماه یا 4 ماه هرچی، آیا معنیش اینه که این آلولا دوباره احیا میشن یا یه اتفاق دیگه؟
بسیار عالی، سوال بسیار خوبی بود آقای دکتر، به نکته خیلی مهمی اشاره کردین. اگر دقت کرده باشین اکثر مریضای کووید موقعی که ما ایمیجینگ ازشون انجام میدیم یه الگویی هست که بهش میگن الگوی گراند گلاس اپیرنس یا اون حالت شیشه مات. این الگو موقعی ایجاد میشه که اونجا التهاب هست ولی التهاب شاید انقدر شدید نباشه که نهایتاً کانسالیدیشن و دنس فیبروسیس ایجاد بکنه، یعنی سلول توی حالتی قرار میگیره که به همین خاطر هم با همین استدلال ما به مریضمون کورتون میدیم، ضد التهاب میدیم که میخوایم از اون فاز التهاب خارجش بکنیم. در حقیقت توی یه ناحیه بینابین قرار میگیره. تلاشی که ما با مصرف داروها و کنترل کردن زمینه میکنیم اینی که نره به سمت فیبروز مطلق. اگر فیبروز شد طبق فرمایش شما دیگه برگشت ناپذیر شاید بشه لیبلش کرد، ولی اکثر مریضا تو همین حالت فیبروز کامل نشده هستن، یعنی یه حالتی هست که اون واترشد اریایی که حالا میتونیم بگیم و وقتی که این کارو میکنیم اکثرا که برمیگردن، یعنی ما مریضایمون که دیگه دنس کانسالیدیشن به هم میزنن عکس هم که بعد میگیریم 6 ماه 1 سال هم که میگیریم یه آثاری ازش باقی میمونه. من همیشه برای بیماران مثال میزنم، میگم موقعی که ما دستمون زخم میشه، موقعی که عمیق نباشه یه خراش باشه، بعد از 15 روز ما 2 ماه کامل دیگه برطرفش اصلا نمیتونیم جای آسیبو پیداش بکنیم، ولی وقتی که دیپ کامل فول تیکنس اسکین باز شده اون جوشگاه زخم و اون اسکار تا سالیان سال امکان داره باقی بمونه. دقیقاً همین اتفاق هم توی ریه ها میفته و تمام تلاش و سعی افرادی که با این مریضا کار میکنن اینه که وارد اون فاز تا حد امکان نشود. ولی فرمایش شما صحیح، اگر دیگه فیبروز استابلیش شد برگشتش به این راحتی به احتمال خیلی زیاد نخواهد بود، ولی اکثر مریضا تو همین فاصله هستن و میشه بهشون کمک کرد.
آقای ببخشید، این مشکلی فرمودید بیشتر از سمت ریه است یا سمت واسکولارش هست، یعنی یا هر دو سمت؟ یعنی آیا در عروق هم مثل حبابچه‌ها عروق ریه همچین اتفاقاتی میفته؟ یعنی اون
به نظر میاد اینطور این اتفاق میفته به خاطری که اون بحث افزایش دیدایمری که ما داریم که بحث حالا لختگی و اینا رو مطرح میکنه، به نظر میاد همزمان یه التهابی توی دیوار عروق هم باشه و این ویروس رو تقریبا میشه گفت هیچ جایی از بدن نمیتونیم بگیم که نسبت بهش امنیت داره و ایمن هست. یعنی با هر پرزنتیشن الان هر روز که شما لیتریچر رو ریویو میکنین یه کیس ریپورت خاص که نمیدونم فلان ارگان فلان عضو مثلا نمیدونم سربلوم درگیر شده یا نمیدونم رتین درگیر شده، جاهای مختلف درگیری اتفاق افتاده و ریه هم در حقیقت جدای از اینا نیست. علاوه بر آلوئول که خب به صورت واضح و مشخص داریمش، توی عروق هم میتونه همین داستان باشه. البته حالا پاتولوژیستا و همکارانی که کارهای در حقیقت اتوپسی این مریضا رو انجام میدن شاید ریپورت‌های بهتری بتونن بدن، ولی نمیتونیم درگیری عروق رو به این راحتی کنار بذاریم. من به نظرم عروق هم باید یه قسمتی از کار باشن و حتما هستن احتمال زیاد.
تشکر میکنم. خیلی ممنون.
خیلی ممنون آقای دکتر. من یه سوال دارم، سوالم کوتاه میپرسم چون وقت شما رو به اتمام هست. چقدر احتمال داره که بیمارانی که مبتلا به کووید نوع شدید بشن دچار پولمونری هایپرتنشن در دراز مدت بشن؟ چقدر این احتمال ممکنه وجود داشته باشه؟ آیا موارد یا شواهدی وجود داره به صورت گزارشی انجام شده یا خیر؟
خانم دکتر، اگر ما بحث آمبولی ریه رو تو این قضیه جدا بذاریم یا باهاش جمع بزنیم یه مقداری متفاوت میشه. ما مریضایمون که خودشون دچار آمبولی ریه میشن بستگی به وسعت و شدت آمبولیه و اینکه منیجمنت رو کی گرفتن، اینا میتونن یه درصد خیلی کمشون امکان داره که در حقیقت نهایتا منجر به سیتف بشه و به صورت کرونیک طرف آمبولی داشته باشه و پولمونری هایپرتنشن به هم بزنه. اون اگر اینا رو تفکیکش بکنیم که یه مقدار کار سختیه، خیلی تفکیک کار راحتی نیست که مریض فول همه استادیا بشه و بگیم خب این PTE نداشته الان پولمونری هایپرتنشن داره، یا اینکه برعکس این باهاش بوده. ما خود مبحث پولمونری هایپرتنشن موقعی که نگاه میکنیم 3-4-5 تا زیر مجموعه براش در نظر میگیره که پارانشیم که مشکل داشته باشه نهایتاً میتونه منجر به پولمونری هایپرتنشن بشه، واسکولار که باشه، مسائل کاردیوواسکولار که باشه، اینا باز میتونه باشه. اینکه عدد مطلق من جای دیده باشم با توجه به دیتایی که الان یه چیز هولوش 1.5 تا 2 سال هست که داره حالا کار میشه، به صورت مطلق عدد درصد داشته باشم من تو ذهنم عدد درصد ندارم، ولی پتانسیالی فرد میتواند به خاطر بحث هایپوکسیا، به خاطر التهاب و به خاطر آمبولی نهایتاً منجر به پولمونری هایپرتنشن برای مریض بشه. ولی این پولمونری هایپرتنشن چقدر موندگار باشه و چقدر ریورسیبل، این بحثیه که خب یه مقداری زمانبندی نیاز داره و نیاز هست که کار بیشتر بشه. آیا مریض حالا با منیجمنت یا بدون منیجمنت چه اتفاقی براش میفته؟ شاید 6 ماه 1 سال دیگه بیاد که بگه همون مریضایی که ایسیمپتوماتیک هم هستن ما اگه فلان دارو رو بهشون بدیم توی لانگ ترم جلو پولمونری هایپرتنشن بعدی اینا گرفته خواهد شد. این هنوز دیتاش نداریم، دیتا رو به صورت مطلق. حالا من دیتای بیشتری در دسترس ندارم که خدمتتون ارائه بکنم.
خیلی ممنون آقای دکتر، خسته نباشید. Okay, I ask Professor Masumpour how often pulmonary hypertension may happen in the patient with severe COVID-19, and he said we don't have enough data up to now, but maybe when hypoxia is with emboli, the prevalence of pulmonary hypertension increases with time. But of course, we don't have enough data. Maybe in the future, we will have this data about the prevalence of pulmonary hypertension in the patient with severe COVID-19. خیلی ممنون، متشکرم. Let's move to the next speaker. It's my pleasure to invite my professor, Dr. Hamid Najafipour, from Kerman University of Medical Sciences. He has so many experiences in cardiovascular and respiratory physiology. He is also advisor of the research chancellor of Kerman University of Medical Sciences. You can start, please.
Okay. Do you have my voice?
Yes, yes.
Okay, thank you. In the name of God, the beneficial, the merciful, it is of great pleasure for me to have a talk in the 4th International and 25th National Physiology and Pharmacology Congress. The title of my talk is Pulmonary Hypertension and Non-Coding RNAs. Pulmonary arterial hypertension is a severe and life-threatening disease with still largely unknown pathogenesis. The development of PH or PAH is multifactorial with genetic background and environmental stresses as two critical components. PAH develops as a result of the pulmonary arteries lumen occlusion. Right heart failure is a common cause of death of patients with severe PAH. Hypoxia is a common cause of PAH. It induces the expression of hypoxia-inducible factor 1, HIF1, that is a transcription factor and regulator of cellular response to hypoxia. Some hypotheses for PAH: the imbalance between endothelium-derived vasodilators and vasoconstrictors, reduction of NO availability or NO production, local inflammation in the vascular wall, imbalance between the pro-inflammatory and anti-inflammatory cytokines, imbalance between the apoptotic and anti-apoptotic proteins, decreased level of P21, that is a cell division and proliferation inhibitor. Let's have a look to what happens to the vascular system of the lung in PAH. In the left side, let me have a pointer. Okay. Do you have my pointer now?
Yes, yes, we have.
Okay. In the left hand, we can see a normal pulmonary artery, small artery or arteriole with 3 layers. The inside is the endothelial layer or intima. In the middle is the media of a smooth muscle layer, and the outer layer is the fibroblastic cells, and it's called adventitial. So due to stresses of 2 pulmonary vessels, or either chemical, biological, bacterial, this causes the endothelial cell dysfunction and proliferation. So the layer of the endothelium is increasing thickness. Smooth muscle cell hyperproliferation and resistance to apoptosis means that the medial layer is increasing thickness again, much more than the endothelium, and also fibroblast activation that leads to increase in the number of fibroblasts and also the thickness of the outer layer. The final result is that the thickness of the arterial wall is tremendously increased, and because it is going toward the inside of the vessel, the lumen width is significantly decreased. As we know that the resistance to flow is inversely related to the radius of the lumen to the power of 4, it means that with reduction of the lumen diameter, there is a huge increase in the pulmonary vascular resistance. So therefore, there is an initiation step with proliferation, inflammation, anti-apoptic, and goes toward the in the progression, and then to remodeling. The final step is remodeling, that is irreversible. But in the middle progression, it is reversible. It depends to the timeline of the progression of the disease. So the genetic variants, age, hormones, shear stress, oxidative stress, drugs, viruses cause the endothelial dysfunction. This leads to vasoconstriction, medial hypertrophy, and cytokine release, inflammatory cell recruitment, and then metabolic shift, medial hyperplasia, ECM remodeling, extracellular matrix remodeling, apoptosis resistance, increased proliferation, finally leads to intimal lesions and plexiform lesions in the endothelial layer, and also thrombosis after that. Adventitial fibrosis, that's the outer layer, and increased arterial wall thickness. So this will lead to right ventricular failure. That is the main cause of death in the PAH patients. This is a X-ray micro-CT image from a model of mice with PH, with familiar PH, a model familiar PH. As you can see, that in the lung we have a large vascular system with many branches of the vascular system. In PH, there's a reduction of the number of vessels. Many vessels are truncated. It means that there's a huge reduction of the number of vessels. With having tortuous vessels, this will the reduction of the number of vessels, because they are parallel in circulation, they will increase the pulmonary resistance. This is along with the thickness of the vessel wall and the reduction of the lumen. All of these will cause a huge increase in pulmonary resistance, in vascular pulmonary resistance. This is a nice slide about the time table or timeline of the changes of the in the lungs due to exposure to hypoxia. As we know that hypoxia is a good reason for PAH. It's by Lee Young 2019. It shows that the rate of 3 weeks of exposure to hypoxia is week 1, week 2, week 3. There's an increase in pulmonary ventricular systolic pressure, increase in systolic pressure linearly with the days, with the weeks, and also increase in mean pulmonary arterial pressure. Here is the right ventricle to left ventricle thickness. It means that right ventricular hypertrophy index is an increase in the hypertrophy index. And here we can see again, it is a vessel, a small artery or arterial in the lung. It's a normal with open lumen and wide lumen, and it is the thickness is not very much, but you can see with increase in the time, there is increasing of the thickness of the wall and decreasing the lumen, and this is the wall thickness as a score is increasing in wall thickness during 3 weeks of the disease. What about non-coding RNAs? We know that about 1.2% of human genome encodes proteins. What about the other 98.8%? The rest is pervasively transcribed into many non-coding RNAs. Non-coding RNAs means the RNAs that not produce protein There are no expression of protein. They have only regulatory effects. Non-coding RNAs are divided by two groups of microRNAs composing of 18 to 25 nucleotides, and long non-coding RNAs composing of more than 200 nucleotides. What about the role of microRNAs in pulmonary hypertension in the heart? Here is a nice slide showing the normal pulmonary vessel and a hypertrophied one. It shows that the thickness of the vessel has increased, and so all the lumen width of the lumen is significantly decreased. All three layers, there is an increase in the thickness of all three layers. It is a list of RNAs, microRNAs that are involved in the proliferation or and thickness of the vessel wall. For example, for the intima, there is MIR 17, 20, 27, 424, and 503. These are involved in the thickness of the intima layer. And there's a very long list of the microRNAs that are involved in the increase in the thickness of the media, vascular smooth muscle cells, proliferation of them, and also some of microRNAs that are involved in the thickness of the outer layer of the vessel. So it means that microRNAs are quite involved in this in the process of changing a normal vessel to a hypertrophic one. Inflammation is a key initiator of the PAH. Here it shows that TNF-alpha, interleukin-6, and PDGF cause DNA damage. One of the reasons for PAH is DNA damage. DNA damage leads to increase in production of a part 1, which is a factor for DNA repair. And also, increase in part 1 cause DNA repair, but it will lead to increase in proliferation and decrease in apoptosis. All of these leads to PAH. This is direct effect of the PAR. The indirect effect of PAR is through the MIR 204, NFAC2, and HEAF. This also leads to proliferation and finally to PAH. Therefore, inflammation through DNA damage directly or indirectly leads to PAH. This is a normal heart. This is a hypertrophic heart, a fibrotic heart. That shows that what happens in PAH to the heart is at first we have to say that microRNAs are involved in the normal heart, in proliferation, in cardiac conduction, in differentiation, in the normal heart. But in the hypertrophic heart, there's many more of them that are involved. This is the normal cardiomyocyte cells, and this is the fibroblastic cells that are increasing in number, and also there's ECM deposition in the between the normal cells. This is called the thickening of the ventricle, and also the stiffness of the ventricle. That is this cause to increase in the pressure, and also will lead to heart failure if it is not cured. So there's some microRNAs here that was listed here that are involving in apoptosis, hypertrophy, myosin switching, and finally fibrosis. Some of them are inhibitory, some of them are excitatory, means that these microRNAs are involved in production of fibrosis, or they are against production of fibrosis. In the vessels, in all phenomena in the vessel that are involved, for example, the MIR21 is involved in proliferation and restenosis of the vessel wall. Is in no intima, that is, it is a increase in the plug formation that reduces the lumen of the vessel. Also in ischemic angiogenesis, in angiogenesis, in arteriosclerosis, in vascular remodeling, and increase in blood pressure through angiotensin system. There are many microRNAs that are involved in this phenomenon. Therefore, both in the heart and also in the vessels, microRNAs are involved in pathologic conditions. What about the long non-coding RNAs? After this discovery of microRNAs, it was later shown that long non-coding RNAs reveal a new layer of regulation in eukaryotic cells. Long coding RNAs can control the flux of genetic information, such as chromosome structure modulation, transcription, splicing, messenger RNA stability, messenger RNA availability, and post-translational modification. Long coding RNAs present interaction domain with DNA, messenger RNA, and proteins. It means as transcriptional, post-transcriptional, and also in translational stages, these are also involved. This is a slide that shows long coding RNA and regulation of gene expression. These are involved in gene expression. My colleague, Dr. Jafari Najad, the last speaker of this session, this symposium, will speak more about the effect of the role of long coding RNAs. Here, just only I will describe a little of them. This is a long coding RNA that it is involved in methylation or process in the nucleus that will lead to repression of genes. This causes repression of genes in cis and also in trans forms, cause repression of genes. Also, there are messenger RNAs that goes to the cytoplasm to express proteins. We know that microRNAs are attached to the messenger RNAs and cause the degradation of them. No, the long non-coding RNAs make a sponge that microRNAs goes to the sponge, and therefore the messenger RNAs are released, and no, they can go toward the expression of proteins. So it means that there is an interrelationship between microRNAs and long non-coding RNAs in expression of proteins. Also, long coding RNAs are involved in the messenger RNA decay or messenger RNA stability. What about our results? We use monocrotaline to induce PAH. It is a chemical factor that injected to the animal just once, so a sub-Q dose. After 3 weeks, it will lead to PAH because it is a very invasive molecule. It is an oxidant. It is an inflammatory molecule that causes PAH. The results in the lung shows that the arteries in the lung increase in the thickness. This is the MCT animal. This is the MCT vehicle animal. It shows that the small artery after 3 weeks is a.
Excuse me.
Sorry.
Excuse me, Professor. Your time is going to be finished.
Okay, just maybe about 3 minutes, 2 or 3 minutes.
Okay.
Okay, that's the narrow lumen and thick wall. This is wall thickness. This is the control group. Is MCT as MCT plus vehicle is an increase in the wall thickness, and also this is a HIF. Is increase in HIF as gene and as protein expression of HIF. This is a MER 204 in the lung. That's a reduction of MER 204, and also in 229A, there's a reduction of the expression of the microRNAs. Part 1 is increased, also in gene and also in protein expression. There is a reduction of SOD, reduction of GPX, reduction of catalase, and in the heart, there is an increase in right ventricular systolic pressure. There is an increase in right ventricular hypertrophy index. There is increase in right ventricular fibrosis. No change in microRNA 27A, but again, is reduction of microRNA 204. Interleukin 6 increase, TNF alpha increased, catalase decreased, TAC decreased, and MDA increased, and also MDA to TAC. It means that the ratio of oxidative stress to antioxidative protection, there's an increase in oxidative stress in the lung. This is BAX, increase in BAX, decrease in BCL2, and the level BCL2 to BAX has decreased in this one. P21 has an increase in P21 and increase in caspase-3. In conclusion, drugs, toxins, shear stress, genetic variants, oxidative stress, virus stress, hormones cause dysregulation of microRNAs, DNA damage, and at least to up to ECM remodeling. This will lead to cytokine release, vascular remodeling, vasoconstriction, and increase in vascular resistance. This will lead to PAH. The direct effect of these factors is increased RV afterload and inflammation, and indirectly through the release of some inflammatory factor from the lung will go toward the right ventricle afterload increase. And finally, there's again a dysregulation of microRNAs and the right ventricular hypertrophy, oxidative stress, and finally right ventricular dilatation and failure. This is the final step in PAH. About long non-coding RNAs in PAH, all 3 layers of the vessel wall will be hypertrophied to produce pulmonary hypertension. It is a vessel with a thick wall and narrow lumen, and long coding RNAs are involving in all of these processes. For example, migration, proliferation, apoptosis, and cell cycle. They are involved. There is many long coding RNAs that are involved here. Many of them are here. Many of them are here. Some of them are excitatory. Some of them inhibitory. So they are also involved in this process. Thank you for your attention.
Professor, thank you very much for your very interesting and great presentation. Unfortunately, we don't have enough time. If audiences have questions, they can ask at the end of our program. Thank you.
Okay, fine.
Now we move to the next talk. It's my pleasure to introduce my professor, Professor Norbert Weizmann, Chair for Molecular Mechanism and Emphysema, Hypoxia and Lung Aging in excellent Glasser Cardiopulmonary Institute in a very famous and advanced lung research center in Gießen, Germany. Hello, Norbert, and welcome to our program. I cordially appreciate for your time and sharing your experiences with us in the program. Thank you. You can start, please.
Yeah, thank you. I hope you can see my presentation and you can hear me clearly.
Yes, we see.
Yeah, so I would like to thank you and the organizers very much for inviting me to give a presentation, and I'm really honored to be part of your Congress. So I will talk about pulmonary hypertension and chronic obstructive pulmonary disease. And of course, you know that COPD, chronic obstructive pulmonary disease, is a major worldwide health problem. Indeed, the WHO prognosis is that it will rank on the third most important cause of death worldwide by 2030. We also know that we have a variety of factors that cause COPD. For instance, genetic predisposition, infections, the socioeconomic state, and aging. But it is clear that air pollution and tobacco smoke are the major causes of COPD. And I guess we all remember images like this that show our air pollution. Before I go deeper into the matter, I would like to give a brief overview about the lung structure and gas exchange. It is clear to all of us that for gas exchange, the lung has to create a very large surface and a very thin diffusion barrier between the gas and the airspace. And this large surface is caused by branching, as indicated here, by the airways and by the vessels. And if I focus on the gas exchange regions, this is a terminal bronchial where the oxygen reaches our lungs alveoli, where the gas exchange occurs. And if I go deeper into the structure, this is an electron microscopic slide. All those structures that are marked with A represent an alveolus where the gas exchange happens. And I hope this gives an impression how the lung creates this big surface. And if we go even deeper, we have reached the gas exchange barrier where the oxygen is taken up from the air and CO2 is released from the blood. This is the capillary. And if I magnify even more, this slide should show you how thin the barrier between the airspace in the lung above here and the gas space and the blood is. This is 2 micrometers, so the barrier can be as thin as 0.2 micrometers. And I guess this makes clear why the structure is so prone to attacks, to environmental toxical agents, to tobacco smoke. This is in comparison, the slide I showed you, to a human hair. So when we talk about COPD, we have to take into account that we have 2 effects in COPD. The first is happening here in the larger and smaller airways, which is the chronic bronchitis, which is increased mucus production, which is inflammation, which leads to airway remodeling as shown here, a healthy airway and a remodeled airway with a narrowed airway lumen that makes breathing more difficult. This also can lead to instability of small airways and collapse of airways, which leads to the phenomenon of air trapping. But then we have a second phenomenon, which is the lung emphysema, which is defined by a destruction of the gas exchange surface of the alveoli. This is what happens down here. And at the end, emphysema results in a complete destruction of the alveoli. In simple words, we have something like holes in our lungs. And I would like to talk about emphysema mainly. The classical view of COPD is therefore that it is an airway disease with the airway remodeling, with all I showed you. But we have learned that COPD is obviously much more than an airway disease. If you think about the muscle wasting that occurs in the patients. And this has brought up the idea that COPD is a vascular disease, and not only a vascular disease of the body also, but especially of the lung. And this field of research was triggered by the research group von Joan Barbera in Spain, who looked at pulmonary vessels of COPD patients that have of smokers that have not yet developed COPD. And this looks, we just have heard about pulmonary hypertension, very much what we are used to see in pulmonary hypertension, a thickened vessel wall and a narrowed vascular lumen. And when we looked in our tissue bank, this is from a healthy human lung, and this slide is from a smoker, we found similar thickening of the vessel walls and narrowing of the lumen. I guess I can skip these slides. We have heard just about pulmonary hypertension, which is induced by a thickening of the vessel wall as shown here, and which leads to an increased afterload for the right heart, which finally results in deterioration of right ventricular function and right heart failure, where patients die from. So when we looked at these images, we saw this, as I said, looks what we are used to see in pulmonary hypertension, and this was new, the occurrence of pulmonary hypertension in smokers that have not yet developed COPD. There is no doubt, as I will show you later, that if you have severe COPD and have hypoxia, via the hypoxia mechanism, there's development of pulmonary hypertension. But these investigations suggested that this is an early phenomenon. And meanwhile, it is clear that moderate to severe pulmonary hypertension is not a rare event in COPD, that it is of prognostic relevance for the patients, but the prevalence in mild and moderate COPD was not known. The new figures show that about up to 70% of COPD patients can even have mild, at least pulmonary hypertension. So this is the background, and there we ask, is this a regular phenomenon? Is there always development of pulmonary hypertension in COPD? And what occurs first, the destruction of the alveoli, the emphysema, or the pulmonary hypertension? So we set up a mouse model where we can expose in these chambers mice to tobacco smokes for 6 hours a day, 5 days a week for up to 8 months. This is really a long-term experiment. And these are the main results when we look for pulmonary hypertension. This is the right ventricular systolic pressure. This is the course of smoke exposure for up to 8 months. We found that pulmonary hypertension was present already after 3 months of smoke exposure. When we looked for emphysema here with the airspace measured histologically, we found that emphysema was only detected after 8 months of smoke exposure. So this is very clear. Pulmonary hypertension occurs largely before emphysema development. And when we looked closer at the vessels, we talked about the thickening of the vessel wall that is shown here after 8 months of smoke exposure compared to a healthy non-smoke exposed mouse. We also found this vascular remodeling, the thickening of the wall that also occurred already after 3 months of smoke exposure as shown here. And also we have, in terms of pulmonary hypertension, a loss of vessels. When we look at the ratio of the number of alveoli and the number of vessels, we found that the ratio increases, which points as at a loss of vessels, a higher loss of vessels compared to the loss of alveoli. So there is something with the vessels. Let's come to the introductory question. Is this vascular remodeling driven by hypoxia? This early pulmonary vascular remodeling is not caused by hypoxia. As I said, there is no doubt if you have severe emphysema and severe COPD that the hypoxia occurring triggers pulmonary hypertension. But here we talk about an early phenomenon, and we showed that neither the oxygen concentration in our smoke exposure chambers nor the arterial PO2 is changed after smoke exposure in the mice. So this pulmonary hypertension is not triggered by hypoxia. We looked for different regulatory pathways. It would be too much to go into detail here. But also when we compare chronic hypoxia to smoke exposure and pulmonary hypertension, we had much different regulatory pathways identified here. So this is obviously something independent. And then we asked for the mechanism of pulmonary hypertension and emphysema development. It has since long been suggested that nitric oxide synthases and O production can play a role here. You know that we have three isoforms of nitric oxide synthase: the neuronal, the inducible INOS, and the endothelial form, the ENOS. And we screened for the expression, and maybe we can have a look here. On the cause of smoke exposure. These are healthy controls. This is after 8 months of smoke exposure, the middle column, and after 8 months of smoke exposure. And we found that this is the protein staining, that the inducible nitric oxide synthase was upregulated, and in particular in the vessel walls. Whereas the other isoform, the endothelial form, was rather downregulated. So this was the first hint that the inducible nitric oxide synthase can play a role here. And then we exposed different genetic mouse lines, wild-type mice. These are the left two bars. The middle bars are from inducible nitric oxide synthase knockout mice, so they don't have this enzyme, and the two right bars are always from the endothelial nitric oxide synthase knockout mice. And when we first looked for pulmonary hypertension, we of course found pulmonary hypertension after 8 months of smoke exposure, very similar in the endothelial knockout mouse. But the INOS knockout mice did not develop pulmonary hypertension, as shown here by the middle bars. We looked for right heart hypertrophy, which also did not occur in INOS knockout mice. And also the vascular remodeling process indicated here was absent in the INOS knockout mice. So there was, after smoke exposure in INOS knockout mice, not any development of pulmonary hypertension. What about emphysema? These mice, and I put here a structural parameter, the so-called mean linear intercept, which shows the distance from one alveolar septal wall to the next, and also a functional parameter. And similar to pulmonary hypertension, only INOS knockout mice, here the two middle bars, were protected from lung emphysema development. We thought, okay, the final goal is to treat patients. We are far away from that, but we treated our mice. We first used an inhibitor of the inducible nitric oxide synthase, the L-NEIL, in parallel with smoke exposure, and similar to the INOS knockout mice, the mice were protected from emphysema and pulmonary hypertension development. And encouraged by these results, we looked for a curative approach. We first exposed the mice to 8 months of smoke, and then we treated them either with placebo or the INOS inhibitor for another 3 months. And to our surprise and to our happiness, this led to curation from lung emphysema and from pulmonary hypertension. So this is the healthy control. This is the placebo-treated mouse. And this is after 3 months with the INOS inhibitor. So this was fantastic. This was lung regeneration. We could cure emphysema, at least in the mice. And if you have such a result, you have to look with different techniques whether this really is true. The most reliable measurement is to count the number of alveoli with design-based stereology. So this is the healthy control. This is placebo-treated mice. They have only half the number of alveoli. And after the curative treatment, the number of alveoli increased. There was really and indeed restructuring of the lung. So we thought about the signaling. INOS produces nitric oxide. This can combine with superoxide to peroxynitride, and we thought peroxynitride is the signaling molecule that does the downstream signaling, and peroxynitride leads to nitration of proteins, nitrotyrosine formation, which is shown here. And in fact, after smoke exposure, nitrotyrosine levels were increased in the smoke-exposed lungs. This is also shown in the immunostaining down here. It also has a functional effect. I would like to show you only one result here, if we use peroxynitrite and apply this to epithelial cells which are isolated in culture, this decreases proliferation and increases apoptosis of epithelial cells. So this really leads to emphysema development, and there are similar effects on endothelial cells. You may ask whether this is also relevant for the human disease. As far as we could investigate, yes, we looked at lung samples from healthy donors, from smokers that have no COPD, and from smokers with COPD. These are the structural changes, the emphysema development, as shown here in the three upper panels. We have, similar to the mice, the increase in the ratio of the number of alveoli to the vessels. And we have the thickening of the vessel wall, the vascular remodeling, similar to the mice. And when we look for the regulation of the inos, maybe we look only on the protein here, the inducible nitric oxide synthase, so it was gradually upregulated in smokers without and with COPD, whereas similar to the mice, the enos was rather downregulated. So this is our concept. We showed that, at least in mice, deletion of the inducible nitric oxide synthase could not only prevent but also restore emphysema and could reduce pulmonary hypertension to the normal. But as to this concept of peroxynitrite formation, you need superoxide. And we then searched for the superoxide source. There are many possible superoxide sources. Mitochondria have been shown to produce reactive oxygen species, xanthine oxidase, and the large family of so-called NADPH oxidases. You may know well the classical form, the so-called NOx2 isoform of the NADPH oxidase. Previously, this was termed GP91-FOX. This is the NADPH oxidase that is active in our immune cells, in macrophages, which are important for the immune system. But the NADPH oxidases are complex enzymes. They don't have only this membrane-bound subunits here, but also a number of cytosolic subunits that have to assemble to form the active complex that can produce superoxide. And during the last 10-15 years, isoforms of these different subunits have been identified, termed NOx1, NOx3, NOx4, NOx5, and the dual oxygenases, and also isoforms of the cytosolic subunit, termed here, for instance, NOx01 or NOxA1, have been identified. So we looked for the expression of all these isoforms in our mouse model. This is a very complex slide, but the take-home message is this is 2 weeks of smoke exposure, 3 months of smoke, and 8 months of smoke. And very consistently, one isoform, the NOx01 isoform, was upregulated during the time course of smoke exposure. So we thought this might be a good target, and we looked then in detail, for instance, for the protein expression by Western blotting after 3 months of smoke exposure and 8 months, and found that indeed the NOx01 subunit of the NADPH oxidase is upregulated. This is also shown in the staining here on the right. Again, we looked for parallels in humans. This is lung tissue from healthy controls, from patients with mild COPD, and from emphysema. And also in emphysema, the NOx01 subunit was largely upregulated and correlated very well with lung function, as shown here. The higher the NOx01 expression in the lungs, the worse is the lung function in patients. And for proof of principle, we then looked again in a knockout mouse. We created a knockout mouse that lacked the NOx01 subunit, and we again repeated the experiment like for the inos knockout mice. We showed that the number of alveoli was reduced in smoke exposure, but in NOx01 knockout mice here after 8 months of smoke exposure, no emphysema occurred, also no changes in lung function. So these mice were protected from emphysema, but not only from emphysema, but also from pulmonary hypertension. This is a measurement of the activity of matrix metalloproteinases that increases in emphysema development, but did not occur in in the NOXO1 knockout mice. This is a marker for apoptosis, which increases in emphysema, but did not increase in the NOXO1 knockout mice. This was measured by a technology termed fluorescent microscopic tomography, where you can use detectors of the apoptosis signal or the MMP activity. But when we then looked for pulmonary hypertension, as I said, the NOXO1 knockout mice were also completely protected from pulmonary hypertension. This is the knockout mouse response. This is the development of pulmonary hypertension in wild-type mice. And this was true not only for the right ventricular systolic pressure, but for all parameters we measured. For instance, the cardiac output that is decreased in pulmonary hypertension stayed at the normal level. We also looked at the vascular wall thickness, the thickening of the vessel walls that occurred in the wild-type mice on smoke exposure was reduced in the NOXO1 knockout mice. And regarding the downstream signaling, as I said, we suggest nitrotyrosine as the key molecule triggering the harmful effects of the tobacco smoke on the vessels and on the lung structure. The nitrotyrosine levels that were increased in smoke exposure stayed at the normal level in the NOXO1 knockout mice. So with that, I would like to conclude that we have suggested a model coming from our experiments in mice, but also from the parallel investigations in human, that the INOS-derived nitric oxide together with superoxide derived from an NADPH oxidase that is dependent on the NOXO1 subunit reacts to peroxynitride, which does, for instance, via protein nitration, the harmful effects to the lung that cause pulmonary hypertension, which then may also trigger the emphysema development. And with that, I would like to thank all of my collaborators in this study and would like to thank you very much for your kind attention and would be happy to have your questions if there are any. Thank you.
Professor Weissman, thank you very much for very excellent talk. I really enjoy, and I'm sure that many audiences agreed with me. If the audiences have any question or comments, their microphone will be on. Also, you can ask your question by chat. Dr. Najafi, please.
Thank you, Dr. Weissman, for your nice talk and very good slides. I have one question about the inhibitors of NOS. The problem with inhibitor of NOS is systemic hypertension. Do you have any experience with using these inhibitors on systemic hypertension or blood pressure?
Yeah, so I agree. You're absolutely right. We know that there had been, especially also regarding ARDS, acute respiratory distress syndrome, and in septic events, clinical trials that used inhibitors of the nitric oxide synthase. And you're absolutely right. That if you inhibit nitric oxide synthase, this can lead to systemic hypertension, but also to pulmonary hypertension. What we found is that specifically the inducible isoform of the nitric oxide synthase is relevant here. So the goal is to only target the inducible nitric oxide synthase and not the endothelial form, which is the major productant of the vasodilatory nitric oxide we have for vasoregulation. So we use this compound termed L-Nil. It has not been developed for clinical application, and you're absolutely right. The trick here is to have an INOS, an inhibitor that only targets the inducible nitric oxide synthase, which is upregulated in smoke exposure. And I would predict that this has then no side effects on blood pressure.
Okay, thank you. The second question is that how you did provide from human lung, a sample from a normal human lung?
Yeah. So that's an excellent question. Of course, we have very tight regulations in Germany and also worldwide using human lung samples. So the healthy samples were from plant lung transplantation, from lungs that have been planned for lung transplantation, but could in rare cases not been transplanted. So if you have, for instance, a traffic accident and the lung is thought to be transplanted, and then it's taken out by the surgeon, and all the ethical approval is there, and then the surgeon recognizes that it was nevertheless damaged in large parts so that it cannot be used for transplantation. So we have access to some of those lungs that we can use for controls.
Thank you very much.
Thank you.
Thank you very much, Professor Weisman. Thank you.
Thank you. Thank you. It was a pleasure.
Hello, everyone. My name is Farzaneh Ketabchi. I'm Associate Professor at Shiraz University of Medical Sciences, Shiraz, Iran. And in this PowerPoint, I would like to give you some information about isolated perfused lung, which has been used for a long time by investigators interested in the physiological, biochemical, and metabolic aspects of the lung. And also this method has been used to study the mechanism of ARDS, ischemia reperfusion injury, pulmonary vascular responses to hypoxia and hypercapnia, and pulmonary hypertension. There are so many advantages for the isolated perfused lung. For example, in this setup, the lung is intact with physiological cell-to-cell contact and native extracellular matrix and cell polarities. And also, in this setup, we could exclude the influences of external factors like the nervous system, plasma-borne mediators, hormones, and so on. It is also possible to use different composition of gases and evaluate the gas exchange and hemodynamic without being worried about the effects of gases in the body. We can monitor the mediator release and metabolic events sequentially in the buffer medium. And finally, we can control the hemodynamic variables and evaluate vascular pressure, vascular resistance, membrane permeability, and fluid balance in this method. However, there are some limitations in the isolated perfused lung. For example, because we have different cell types into the lung, therefore, any physiological and biochemical events cannot be ascribed to just one cell type. And also, we cannot increase the time course of experiments more than a few hours. And finally, even during a stable condition, we cannot exclude any activation of biochemical and metabolic pathways in the system. Taken together, the advantages of the isolated lung is much more than the limitation of it. Therefore, many scientists use this setup to study the vascular responses to pharmacological agents and different composition of gases, and also the mechanism of pulmonary hypertension. The isolated perfused lung preparation has been obtained from almost any experimental animals, given that we have some limitations in using large animals because of ethical and economical issues. Therefore, many scientists prefer to use small animals like rabbit, rat, and mouse. It is important to mention that we must use very well developed and updated devices in the isolated setup to get a reliable results. In general, in the isolated lung setup, we have an external circulation in order to regulate the temperature of the perfused and the lung, and internal circulation to have a perfusion flow into the lung, and ventilator and different composition of gases to have a ventilated lung and normal pH during the experiments. One of the modified and well-designed setup has been established by Professor Werner Seeger and their colleagues in Gießen, Germany. In this setup, the suspended lung is located in the humidified chamber and connected to a force transducer to measure the lung weight, which is very important factor in this setup, because the lung must be isogravimetric during a stable condition, and also in some maneuvers like hydrostatic challenges, in order to measure pulmonary capillary filtration coefficient, it is very important to measure weight gain by a force transducer continuously when we increase the height of the venous outflow. The pulmonary artery and left atrium also must be cannulated. In both pulmonary artery and left atrial cannula, there are two inner cannula, and these inner cannula are connected to pressure transducer, and then we can measure continuously the pressure of pulmonary artery and left atrium throughout the experiments. And also, the cannula of trachea is connected to a pressure transducer, and therefore we can measure the pressure of airways continuously during our experiments, which is very important variable to evaluate pulmonary edema, because the pressure of airway increases during pulmonary edema. In this setup, there is a closed circulation, which means that the perfused solution into the reservoirs is transferred through peristaltic pump to the pulmonary artery and the lung. Then the venous outflow is transferred back to the reservoirs. The height of the venous outflow is adjusted to have a zone 3 blood flow in all parts of the lung. There is a blood filter and bubble trapper on the way of the perfused flow in order to prevent any interruption of the perfused flow by clot or air bubbles. A positive end expiratory pressure of 1 centimeter of water is chosen to prevent alveolar collapse. There are different compositions of gases which can be used based on the experiment. We can also measure NO released from the expired gas by a chemiluminescence NO gas analyzer. This slide is showing the isolated lung setup in our lab. As you can see, here is humidified chamber, 2 reservoirs, peristaltic pump, bubble trapper, many tubings, circulator, ventilator, and different compositions of gases, and recording system, and so on. We can use different composition of gases, depends on our experiments, but of course we must have a normoxic gas in all experiments to have a normal pH. A normoxic gas is composed of 21% of oxygen. Sometimes in some experiments, scientists prefer to have 16% of oxygen and 5.3% of carbon dioxide, which is balanced with nitrogen gas. We may also use different composition of perfused solutions, even without any oncotic agent, especially if we want to have experiment in a short time. However, in longer experiments or in the experiments of hydrostatic challenges, we must use an oncotic agent such as 5% of hydroxyethyl amylopectin with molecular weight of 200,000 or 1 to 4% of albumin or dextran with molecular weight of 70,000. Sometimes we may use 10 to 15% of plasma or even blood. Having said that, blood is not recommended more often because of some ethical problems in using heterologous blood, and also the cleaning the setup after the experiment will be difficult by blood. I have to emphasize that lung preparation in this setup is very important, otherwise we cannot have a reliable result. At the beginning of a lung preparation, we have to anesthetize the animals with anesthetic agents such as pentobarbital, ketamine, and xylazine, and so on. And then all animals are heparinized with heparin 1000 units per kilogram body weight. Tracheostomy is performed and the chest wall is open with caution. After removing the thymus, we have to separate pulmonary artery from aorta and then cannulate pulmonary artery. Immediately we have to cut the apex of the heart to have a unidirectional circulation with a cold perfused solution. At the beginning of preparation, the temperature of the perfused must be very low, about 4 degrees centigrade, to decrease the metabolism of the lung. Immediately after unidirectional circulation, we have to change the ventilation gas from air to the normoxic gas in order to have a normal pH. In the next step, we have to remove the lung from the chest, but it should be performed with caution without any damage to the lung. Otherwise, we will have lung edema. After removing the ventilated perfused lung from the chest wall, we have to suspend the preparation on the stand and cannulate the left atrium with caution. And then we have to transfer the prepared lung to the humidified chamber and connect the left atrium catheter to the venous outflow. And after that, we have to increase the flow rate and temperature because the flow rate at the beginning of preparation is very low as possible for each setup, and temperature, as I said, is very low. Then we have to increase the flow rate and temperature near to in vivo condition. And in this video, you can follow the summary of lung preparation in the rapid after anesthesia, tracheostomy is performed, the chest will be open, we must open the chest with caution. Diaphragm is cut. In both sides. Connective tissues are separated between the chest wall and heart and lung. Open the chest. Remove the thymus. Separate pulmonary artery and aorta. Cannulate the pulmonary artery. Cut the apex of the heart. Remove the ventilated lung from the chest wall. cannulation of left atrium, and transferring the preparation to the humidified chamber. In this slide, you can see the isolated lung, which is suspended in the humidified chamber. Which variable we can measure in the isolated perfused lung setup? We can measure pulmonary artery pressure, left atrial pressure, ventilation pressure, lung weight, capillary filtration coefficient, vascular compliance, water retention, exhale NO, and some molecular mediators in the perfused solution. This slide is indicating the pulmonary vascular response to alveolar hypoxia. When the lung is ventilated with 3% of oxygen, the pulmonary artery pressure increases, and when the ventilated gas is changed with normoxic gas, again the pulmonary artery pressure returns back to the baseline level. In some experiments, this maneuver is repeated, or the time course of the experiment increases to have a similar condition in the patient with intermittent or continuous hypoxia. This slide is showing the result of hydrostatic challenges. As I mentioned, we can increase the height of the venous outflow and therefore left atrial, pulmonary vein, and capillary pressures. Then the weight gain is calculated by a special formula to measure capillary filtration coefficient. For example, in the case of ischemia reperfusion injury, the capillary filtration coefficient increases due to the damage in the pulmonary capillary endothelium. Also, there are many pharmacological agents which can be evaluated by this setup. For instance, hemodynamic alterations, vascular damage, and endothelial absorption can be evaluated in this setup. At the end of my lecture, I wish all the best with health and happiness in all your life, and thank you very much for your attention.
Okay, thanks all audiences to be with us. If you have any question or comments about the lecture of the isolated lung, please ask after the last lecture because we don't have enough time. Thank you. Now, to the next talk, I would like to invite Dr. Jafari-Nejad from Kerman University of Medical Sciences. Dr. Jafari-Nejad is interested to share her research with us about some therapeutic approaches for pulmonary hypertension. Dr. Jafari-Nejad, please start.
Hello, everybody. Do you have my slides?
Yes.
Thank you. My name is Saideh Jafari-Nejad. I'm a molecular biologist from Physiology Research Center at Kerman University of Medical Sciences. My project is about peryl alcohol and quercetin, which modulate the expression of non-coding RNase H19 and microRNA 29A in pulmonary artery hypertension in rats. Pulmonary PAH is a multifactorial disease and it initiates with endothelial dysfunction and vasoconstriction. At the progression level, medial hypertrophy, mitogens, and cytokine release, inflammatory cell recruitment occurs, which is reversible. And at the remodeling stage, metabolic shift, phenotypic stitching, medial hyperplasia, and ECM remodeling happens, which are irreversible, which leads to intimal lesions, plexiform lesions, arterial thrombosis, adventitial fibrosis, and finally leads to right ventricular failure. Pyrrolyl alcohol and quercetin are two plant derivatives. Pyrrolyl alcohol is a monotryptan and quercetin is a flavonoid. You can find them in a wide variety of plants, including red onion, including vegetables. You can see them in green tea, in grains, in leaves, and citrus fruits. They are well known for their anti-tumor activity, and their anti-apoptotic, anti-inflammatory, and anti-proliferative activities have been investigated in cancer. Professor Najafipour with his colleagues have investigated on the anti-remodeling, antioxidant, and anti-inflammatory effects of pyrrolyl alcohol and quercetin in monocrotaline-induced PAH in rats. According to their experiments, they induced the PAH with the injection of a single dose of MCT, 60 milligram per kilogram of MCT, and for the control groups, they injected saline. After 3 weeks, when the PAH developed, they started the treatments. In the MCT plus vehicle group, they injected 5% of ethanol, and in the MCT plus PA group, they injected daily for 2 weeks, 50 milligram per kilogram of pyrrolyl alcohol. And in the MCT plus quercetin groups, they injected daily 30 milligram per kilogram of quercetin for 3 weeks. After that, they checked their results. According to the Masson serum staining results, you can see collagen deposition in pulmonary interstitium in MCT groups, which was significantly reduced after administration of pyrrolyl alcohol and quercetin. They also evaluated the pulmonary arterial wall thickness. As you see, the wall thickness significantly increased in MCT groups, and after administration of pyrrolyl alcohol and quercetin, the wall thickness significantly reduced, and the wall thickness was not significant in compared to the control group. The aim of our study was to investigate if the beneficial effects of pyrrolyl alcohol and quercetin is mediated through modulation of the expression of long non-coding RNAs, especially H19, and miRNAs, especially microRNA 29A. What are long non-coding RNAs? You know that coding regions of the genome is divided into 2 parts. One part of the genome which codes for proteins, and the other parts which codes for RNAs, which are not translated into proteins. Non-coding RNAs are divided into 2 classes: housekeeping non-coding RNAs, which include ribosomal RNAs, tRNAs, small nuclear RNAs, and small nucleolar RNAs. And the regulatory RNAs are divided into 2 groups: short non-coding RNAs, which are less than 200 nucleotides, and long non-coding RNAs, which are more than 200 nucleotides in their length. Most of the LNCs are transcribed with DNA polymerase II, and you can find long non-coding RNAs at the antisense strand of DNA. The long non-coding RNAs may be at the intronic regions of a genome. They may be intergenic. They may be located between the coding regions. They may be divergent, or sometimes they are translated from the regions that are close to enhancer of an RNA. You can find LNCs at the nucleus and at the cytoplasm. At the nucleus, LNCs may bind to transcription factors and may activate or inhibit the activity of transcription factors. Sometimes they bind to proteins which are involved in chromatin modifications. And sometimes they are responsible to bring genome parts which are distant. They are responsible to bring them in close. And for example, they bring the enhancer close to the promoter and promote the transcription of the downstream gene. At the cytoplasm, LNCs may activate or inhibit the translation of proteins. In some cases, long non-coding RNAs binds to different proteins and act as a scaffold to bind to different proteins which are involved in a certain signaling pathway. In other cases, long non-coding RNAs binds to miRNAs and prevent them from binding to their targets. In other words, LNCs sponges miRNAs and prevent their activity. According to the RNA Central, the non-coding RNA sequence database, there are about 400 LNCs in Homo sapiens, 100 LNCs in mouse, and about 30 LNCs in Ratus norvegicus. And among the non-coding RNAs, long non-coding RNAs occupy the largest proportion. About 70% of non-coding RNAs are LNCs, and only 8% of non-coding RNAs are miRNAs. Long LNCs are poorly conserved. If you align the sequences of an LNC, the identity of sequences in most cases will be less than 50%. Here we align the sequence of H19 from human, mouse, and rat. We aligned the sequences and found that H19 shows only 27 identity between human, mouse, and rat. It seems that the activity of LNCs is depend on their secondary structures. The secondary structure of LNCs is more important than their sequences for their function. H19 is one of the first discovered LNCs in 1986 before Human Genome Project. H19 is a maternally expressed gene which has a highly conserved secondary structure. H19 is highly expressed in vertebrate embryo development, but is downregulated in most adult tissues after birth. It often plays critical roles in process of tumor metastasis and progression. Furthermore, H19 is related to other important physiological processes such as hypoxia, metabolism, and oxidative stress. Additionally, some studies have indicated that H19 may be associated with inflammation. Here is a list of LNCs which are involved in cardiovascular development and disease. And you can see that H19 have upregulated in coronary artery diseases, in myocardial infarction and ischemia, in hypertension and heart failure. You can see H19 with high expression in all these kinds of cardiovascular diseases. Liu and his colleagues investigated the relationship between H19 and pulmonary fibrosis. They demonstrated that H19 was a direct target of microRNA 196 and was associated with the expression of collagen type 1, which leads to the fibrosis. They suggested that LNC H19 has a promotive effect on bleomycin-induced IPF, and its function as a molecular sponge of miRNA 196 provides a novel therapeutic target for IPF. There are two studies that investigated the relationship between H19 and PAH. At the first study, Sue and colleagues focused on the inflammatory role of H19 in PAH. They treated the pulmonary artery smooth muscle cells with the pro-inflammatory PDGF, and they found that it increases the expression of H19, which inhibits the microRNA LED7B. And in return, LED7B cannot inhibit angiotensin type 1 receptor, which in return increases the proliferation of PASMC and cause the PAH. In the other study, they focused on the protective effect of melatonin on PAH. They found that injection of melatonin will increase the expression of H19, which in return bind directly to the microRNA 200 and prevent it from binding to its target, programmed dead cell death 4, which is responsible for the apoptosis of pulmonary artery smooth muscle cells. And so they increase the rate of apoptosis, they decrease the rate of proliferation of PASMC cells, and the remodeling of pulmonary artery decreased and PAH improved. Here we use RNA interactome database to find all the direct targets of the long non-coding RNA H19 and found that H19 binds directly to microRNA 29A. You can see the binding site of H19 to microRNA 29A. At the next step, we performed gene ontology enrichment analysis for the targets of microRNA 29A and found that most of the targets are involved in the assembly of collagen fibrils and other multimeric structures, and they are involved in cross-linking of collagen fibrils. They are involved in cell matrix adhesion. You know that microRNA 29 is the most famous antifibrotic miRNA, and which target more than 20 collagen genes. It targets fibronectin and matrix metalloproteinase, which are involved in extracellular matrix turnover and structure. Here you can see our results. You can see when we treated our animals with MCT, the expression level of H19 increased significantly in compared to the control group. After administration of peryl alcohol and quercetin, the expression of H19 significantly decreased. At the next step, you can see that the expression of microRNA 29 is significantly lower than after administration of MCT, but after injection of peryl alcohol and quercetin, you can see the increase in the expression of microRNA 29. We also evaluated the expression of collagen type 1, VEGF-A, vascular endothelial growth factor A, and alpha smooth muscle actin, which are involved in the pathogenesis of PAH. In the MCT groups, the expression of collagen, VEGF-A, and alpha SMA was significantly higher than the control group. Administration of peryl alcohol and quercetin significantly reduced the expression of these genes, which here we can see the protective effects of peryl alcohol and quercetin on the expression of these genes. In summary, we induced PAH with MCT. We observed that the expression of H19 increased, and as we predicted, H19 will bind to miRNA 29 and increase and inhibit its expression. At the next step, microRNA 29 cannot bind to its target genes, collagen, VEGF-A, and alpha SMA, so the expression of these genes will be increased and the PAH will be progressed. After treatment with prenyl alcohol and quercetin, we saw that the expression of H19 decreased significantly, so it couldn't bind to its target microRNA 29A. So the expression of this microRNA increased and bound to its targets, collagen, VEGF-A, and alpha SMA, and prevent them from participating in the PAH progression. So the pathogenesis of PAH and the progression of PAH inhibited. I should note that we are at the start point. Our project is at the start point, and we should do more detailed molecular analysis to clarify the exact relationship between these long non-coding RNAs and this mRNA and the target genes. And we also should correlate the expression of these genes to each clinical features of the PAH to complete our projects. Thank you for your attention. If you have any question, I will answer.
Thank you very much for your great presentation. If the audiences have any question or comments, they can ask by chat. Or they can ask directly by microphone. Thank you very much. So organizer told me that the time of the program is going to be finished. Just to make a conclusion, alveolar and arterial hypoxia is the important reason behind pulmonary hypertension. It may occur in the patient with COPD or in other respiratory disorders such as COVID-19. In this symposium, some molecular mechanism and treatments have been updated. Thanks all speakers and participants in taking part in the program, and excellent lectures of speakers and updating us about the last data in hypoxic pulmonary vasoconstriction and hypertension. Now the program is going to be finished.